So this is the video where we will learn about how activation works and how T cells really see the world. And it's important because this really finishes your picture of the adaptive immune system. So there are going to be two videos that kind of finish the adaptive immune system for you. And really put it all together so you can describe the whole immune response. First, in this video, I want to review lymphocyte development and then show you how part of it works, activation, how part of getting the immune response going, how that works. And I think in the next video we'll see what MHC is. So for now, let's review lymphocyte development. This is how the immune system is controlled, and I'm going to list the steps, but also I'm going to list what we call the cells at the various steps. Because I know if you just see the steps on their individual slides and you see the different names for the cells, it's hard to put it together, but you can put it together and you can understand this and you can remember it. It's going to help you to see it this way and then go back and find it on the individual slides. We start with immature lymphocytes, but actually we start before that with cells we don't talk about much, but they're the ones that are shuffling their DNA to create their randomly generated lymphocyte receptor. Okay? That's what happens first. We are assuming we just ignore that and we start with immature lymphocytes. Immature lymphocytes have not yet gone through education. And so they go through that, but 95% of them are killed. The ones that survive become what we call mature. They're no longer immature, now they are mature, and that signifies they've gone through education. But they are naive. Naive means they have never seen their epitope. They've never seen anything that their lymphocyte receptors can bind to. So they are naive in that way. And their whole job is to wait for activation. These are the ones that are in your lymphatic tissues. These are the ones that are just waiting. This is the giant pool of lymphocytes, where if we can find the right one, we can have a powerful immune response that can protect us from a pathogen. So that's mature but naive lymphocytes. These are the ones that do primary activation. And so if one of them does primary activation, it will become an activated lymphocyte. We don't talk about this much because this is a temporary step. Because what it's going to do immediately is go through clonal expansion. So it's going to divide, and its daughters are going to divide. And they're going to keep dividing, and we won't be able to tell which one was the original. It will have split in half, 20 times or something like that. So the daughter cells are going to go through differentiation, and those are going to be either effector cells, which do the immune system jobs, like make antibodies or kill infected cells, or there'll be memory cells that will wait maybe decades to go through secondary activation. And so when I say that your mature but naive lymphocytes are the ones filling up your lymphoid tissues, or I can't remember if it's lymphatic or lymphoid. Let me know. I don't care that much. But like in your lymph nodes and in your mucosa-associated lymphoid tissue and in your spleen, those are big places where lymphocytes hang out and wait. And so some of them are mature but naive lymphocytes and others are memory cells. Indeed, there are places where there are a lot of memory cells. But the difference is these are the ones that are waiting for a new infection we've never seen before, and these are the ones waiting to be reinfected by something we've seen before. So that's lymphocyte development. So now we can get into the next topic, and that's going to be activation. So we're going to see, if we just go back to the title, we're going to see how activation of lymphocytes works. And that's going to mean showing a mature but naive lymphocyte, showing it its epitope for the first time. That's what's going to activate it. And if you think about it, The first time that epitope exists in the body is when we are infected with a pathogen. That's the very first time that foreign epitope is in the body. So we have to get that to the lymphocyte. And so what you're going to see is that that's always going to involve the major histocompatibility complex, types 1 and 2. So you're going to have to see what those do and really visualize how MHC presents those epitopes to lymphocytes and how lymphocytes look at them. And you'll see it does get confusing because situations where cells look at MHC are involved both in activation and in effector function. So you have to kind of pay close attention to what is happening in all of these situations. There are 5 different situations we are going to see that involve MHC. So first, let's look at activation. So what happens for a T cell to activate? This might be a good time to remind yourself, what are the outcomes of activation? And what are the T cell effector cells? Those are good things to be able to look up quickly. So what happens is a mature, naive T cell is ready to be activated, and when an antigen-presenting cell shows it the right epitope, it shows it the one it can see, the one it can recognize, which is insanely rare. What are the chances that an antigen-presenting cell has the right epitope for a lymphocyte? It's astronomically unlikely, but it happens. So, if an antigen-presenting cell brings the right epitope to a mature naive T cell, it's going to get activated. Antigen-presenting cells show these antigens. They show T cells their antigens using MHC, or the major histocompatibility complex. There are 2 different systems that we will look at, Mhc1 and Mhc2, and their job is to display proteins. And the only way a T cell receptor can look at a protein is if it's on MHC. The 2 different systems, Mhc1 and Mhc2, we'll look at here. So first, all nucleated cells display MHC1, and they use it to show random proteins they find in their cytoplasm to CD8 positive cytotoxic T cells. So remember, these are the cells that kill abnormal host cells. So all of our cells are routinely taking proteins randomly selected from their cytoplasm and putting them on MHC1 and showing them to cytotoxic T cells. All nucleated host cells. So this is every cell except your except red blood cells, really. Erythrocytes don't have nuclei, but most other cells do. So all nucleated host cells can do this. And what they're asking is, I found this protein inside me. Is this the one you're looking for? Because a cytotoxic T cell can only see one thing. It's a lymphocyte. It only has one receptor. It has 100,000 copies of it, but it's just one receptor. And so it's only going to do anything if this nucleated host cell shows it the one thing it can see. And so the nucleated host cell is asking, I found this protein in me, does it match what you're looking for? If the answer is no, which is most likely, then the cytotoxic T cell will keep moving and they'll just ignore each other. But if the answer is yes, and it's what the cytotoxic T cell is looking for, cytotoxic T cell will kill the host cell. That's MHC1. It's used to show random cytoplasmic proteins to CD8 positive cytotoxic T cells. Mhc2 is more specialized, and the only cells that use it to display things they've brought in through phagocytosis or some other endocytosis. And so the cells that use this are B cells, actually. We'll get into that. Dendritic cells and macrophages. And what they are asking when they display proteins on MHC2, what they're saying is, well, I ate this. Is this what you're looking for? I brought this in through phagocytosis. Is this the thing you're looking for? They're going to ask that to a helper T cell. And again, chances are the answer is no. But if the answer is yes, then that helper T cell is going to do whatever its job is. So helper T cells are effector cells. They do a job, but they only do it if they recognize their one protein. So here's a way to think about it. Dendritic cells live in your tissues. You have special ones that live in your skin. And if a bunch of bacteria end up in your dermis, which is a bad situation, they will sample that. And their job is to then move through your lymphatic system to the nearest lymph node, where you have a huge number of T cells, and they will display the proteins from those bacteria to T cells in the lymph nodes. Here are T cells in a lymph node, and each little dark dot is a lymphocyte. It's a T lymphocyte. And so, which one gets activated? Well, remember, each one has 100,000 copies of its T cell receptor. And when a dendritic cell moves through, it very quickly shows lots of different T cells, its epitopes, as quickly as it can. So here we are, T cell activation. So that's how helper T cells and cytotoxic T cells get activated. B cells, it's a totally different thing, and I want you to stop for a second and accept that B cells are activated in a different way. B cells first have to bump into their epitope. Remember, their B cell receptors are basically antibodies sticking out of their cells, and so if they bump into their epitope using their B cell receptor, they pull the B cell receptor in through phagocytosis, and they digest it, and they display the epitope on Mhc2 to a different helper T cell, a type 2 helper T cell. And so they're going to wander around showing it to different helper T cells until they find the right one. And then if they find the right type 2 helper T cell, it will give the B cell the activate yourself signal. And if it never does find the right helper T cell, it will never go through that full activation. And then once that happens, it's activated. So it goes through clonal expansion and its daughters differentiate. So B cells activation, instead of requiring a dendritic cell to bring it the epitope, it actually binds its own epitope and then shows it to a helper T cell, or to many helper T cells. And if one of those helper T cells recognizes it, that helper T cell will give it the activation signal. I think, honestly, this is enough for one video. In the next video, I will show you some diagrams of cells interacting with each other. And that's heavy, and I want you to draw those interactions, and we have to look at the details of them. So let's get into that in the next video, and that's where you'll see MHC and the T cell receptors and co-receptors all working together. So we'll get into that in the next video, and I will see you there.